Welcome to Fundamentals of Biomolecules. Today, we are diving into the fascinating world of amino acids, the fundamental building blocks of life. Imagine trying to build a massive, complex Lego castle. You need hundreds of individual bricks snapped together in a very specific order. In the human body, proteins are those magnificent castles, doing everything from digesting food to fighting off viruses. Without amino acids, life as we know it simply could not exist. Let us begin our journey into understanding their structure. Now that we understand the importance of proteins, let us look at what they are made of. Proteins are responsible for almost all the heavy lifting in your body. But to understand the whole, we must first understand the parts. An amino acid is a small organic molecule that serves as the fundamental structural unit of these proteins. By understanding the shape and structure of a single amino acid, we unlock the secret to how massive proteins fold, function, and ultimately keep us alive. Building on that foundation, let us examine the anatomy of these molecules. At the very center of an amino acid sits a single carbon atom, known as the alpha carbon. This acts as a structural anchor. Attached to this central alpha carbon are four distinct chemical groups. Three of these are always the same: a basic amino group, an acidic carboxyl group, and a simple hydrogen atom. The fourth is a unique side chain called the R group. It is this R group that varies, giving each of the 20 standard amino acids its own unique chemical personality. Knowing the components, we can now write this out chemically. This formula represents the general structure of an amino acid. The NH2 represents the amino group, which acts as a base and can accept a proton. The C-alpha is our central anchor, holding everything together. The R represents the variable side chain, the only part that changes between different amino acids. Finally, the COOH represents the carboxyl group, which acts as an acid and can donate a proton. If the environment becomes more acidic or basic, these groups will gain or lose protons, changing the molecule's overall charge. Before we move on to complex molecules, let us see this with a concrete example. We will analyze the simplest amino acid, glycine. We are given the information that its R group is just a single hydrogen atom. We want to determine its structure and see if its central carbon is chiral. We start with our general formula and substitute the generic R group placeholder with a specific hydrogen atom. This gives us the complete chemical structure for glycine. This simple substitution is our first step in understanding how different side chains alter the fundamental anatomy of the molecule. Now that we have the structure of glycine, let us evaluate its chirality. A chiral center requires the central carbon to be attached to four completely different chemical groups. Looking closely at our glycine molecule, we see it is attached to an amino group, a carboxyl group, and two identical hydrogen atoms. Because it is attached to two identical groups rather than four unique ones, it fails the test for chirality. Therefore, glycine is not a chiral molecule. Its unique symmetry makes it the smallest and most flexible amino acid, allowing it to fit into very tight spaces within folded proteins. Having seen a symmetrical molecule, let us look at one that introduces asymmetry. For our second example, we will identify the structure of alanine. We are given the chemical fact that the R group of alanine is a methyl group, represented by CH3. We begin by writing out the general amino acid formula once again. Then, we substitute the generic R group placeholder with the specific methyl group. This gives us the complete structure of alanine. Understanding this slightly larger molecule helps us understand the vast majority of other amino acids that share this asymmetrical property. With the structure of alanine established, let us test it for chirality. We evaluate the 4 chemical groups attached to the central alpha carbon: an amino group, a carboxyl group, a single hydrogen atom, and a methyl group. When we compare these 4 attachments, we can clearly see that they are all completely different from one another. There are no duplicates. Because the central alpha carbon is bonded to 4 entirely different chemical groups, alanine is definitively a chiral molecule. This means it can exist in 2 non-superimposable mirror image forms, known as L-alanine and D-alanine. To reinforce what we have just learned and proven, let us look at a visual representation. Here you see the general structure of an amino acid laid out clearly. In the center, labeled in black, is the alpha carbon. Branching off horizontally are the amino group on the left and the carboxyl group on the right. Pointing upwards is the standard hydrogen atom. Pointing downwards, highlighted in blue, is the variable R group. This cross-like schematic perfectly illustrates how the alpha carbon acts as the central hub. Every standard amino acid shares this exact backbone, differing only in that bottom blue R group. Now that we understand the basic structure, we need a way to organize these 20 different molecules. Imagine you are organizing a massive wardrobe filled with different types of clothing. You would sort them into categories like winter coats or summer wear based on their properties. In biochemistry, we do the exact same thing. Because the R group is the only part that changes, we classify amino acids based entirely on the chemical personality of this side chain. By grouping them this way, we can easily predict how they will behave when they are strung together to form a complex protein. Building on this wardrobe analogy, let us define our specific categories at a physiological pH of 7.4. The first group is the nonpolar amino acids, which are hydrophobic, meaning they fear water and hide inside proteins. The second group is the polar uncharged amino acids, which are hydrophilic, love water, and form hydrogen bonds. The third group consists of acidic amino acids, which carry a negative charge due to a second carboxyl group. Finally, the fourth group consists of basic amino acids, which carry a positive charge due to an extra amino group in their side chain. To truly understand these acidic and basic categories, we must look at how their electrical charges are calculated. This formula allows us to determine the total net charge of an amino acid. The symbol Z represents the total net charge of the entire molecule. Qa represents the charge from the alpha amino group, which is usually +1 at neutral pH. Qc represents the charge from the alpha carboxyl group, usually -1. Finally, Qr represents the charge of the side chain. If the environment becomes more acidic, groups will gain protons, and the overall charge Z will become more positive. Let us apply this formula to a specific molecule to see how it works in practice. We will calculate the net charge of glutamic acid at a physiological pH of 7.4. We are given the information that its R group contains a carboxyl group. We will use our net charge formula to solve this systematically. We set up the equation by substituting the standard charges for the backbone and the specific charge for the R group. Adding these together gives us a final calculated net charge of -1. This negative charge is highly functionally important in biology. With our calculation complete, let us walk through the chemical reasoning behind each of those numbers. The value +1 comes from the backbone amino group, which holds onto an extra proton at this pH. The first -1 comes from the backbone carboxyl group, which has lost its proton. The second -1 comes from the R group, which also contains a deprotonated carboxyl group. When we analyze this, we see the positive and negative charges of the backbone perfectly cancel each other out. This proves why the side chain entirely dictates the molecule's acidic classification. Having seen an acidic amino acid, let us now calculate the net charge for a basic one. For our final example, we will look at lysine at a physiological pH of 7.4. We are given the fact that its R group contains an extra amino group. We will once again apply the standard net charge formula. We substitute the standard backbone charges and then add the specific charge contributed by lysine's unique R group. Summing these values gives us a final net charge of +1, demonstrating how positive charges are introduced into protein structures. Let us break down the reasoning behind this positive result. The first +1 represents the protonated backbone amino group, which has accepted a proton. The -1 represents the deprotonated backbone carboxyl group, which has given up its proton. The final +1 represents the extra amino group in the side chain, which is also protonated at this neutral pH. Just like before, the backbone charges cancel out. This proves that the positive charge is contributed entirely by the side chain, making lysine an excellent tool for binding to negatively charged molecules like DNA. To bring all of this classification theory together, let us examine this visual chart. The diagram is divided into 4 quadrants, representing the 4 main categories of amino acids. In the top left, we see alanine, representing the nonpolar hydrophobic group with its simple methyl side chain. In the top right is serine, representing the polar uncharged group. On the bottom left, we see glutamic acid, highlighted to show its negatively charged acidic side chain. Finally, on the bottom right is lysine, displaying its positively charged basic side chain. This chart perfectly summarizes how the R group dictates the molecule's category. We have covered a lot of ground today, so let us review our most important ideas. First, every standard amino acid shares a common backbone consisting of an alpha carbon, an amino group, and a carboxyl group. Second, it is the unique R group that determines whether the molecule is chiral and dictates its classification as nonpolar, polar, acidic, or basic. Finally, this classification is not just trivia. It is the foundational step in predicting how a linear string of amino acids will fold into a functional 3-dimensional protein. You now possess the tools to understand these vital molecules. Great work!